Welcome to the second last webinar of the Botanic Gardens Biosecurity Network seminar series. Welcome to all of our online viewers. I'm Daniela Carnval. I'm from Plant Health Australia and the Botanic Gardens Biosecurity Network. This webinar series is being coordinated by the Botanic Gardens Biosecurity Network. The network is coordinated by Plant Health Australia with support from expert collaborators. And the project is funded by the Australian Government's Agricultural Competitiveness White Paper, the government's plan for stronger farms and a stronger economy. Now, before we get too far ahead in this webinar, I'd just like to run through some webinar housekeeping. So today, all of our attendees will be on mute. However, we really encourage you to use the chat box function to ask your questions to individual panelists or to everyone throughout the webinar. What we'll be doing is we'll be taking those questions throughout the webinar, and hopefully we'll have some time at the end of the webinar to have a bit of a Q&A session with our presenter. If we don't get to answer some questions that come through throughout the webinar, what we'll do is we'll take those questions on and we'll respond to those and provide a written response to all attendees when we distribute the webinar recording. So with that, the webinar is being recorded and will be available at the Botanic Gardens Biosecurity Network website, as well as the Plant Health Australia YouTube channel. Now let's get straight into today's webinar. Today we'll be having a presentation from Philip Hurle, who is the Horticultural Manager at the Australian National Botanic Gardens, and he's going to be talking about living with Amilaria and looking at it from a Botanic Gardens perspective. So with that, I won't keep talking too much. I will hand straight over to Phil, who will give you a full rundown on living with Amilaria. Thanks, Phil.
Yes, hello, welcome. I'll start with a brief about the Australian National Botanic Gardens, just if you don't know where we are. We're situated in the eastern slopes of Black Mountain in Canberra in the ACT. We've got an elevation of about 570, 680 metres and have an average rainfall of 660 millimetres a year. Summer temperatures can get quite hot, average of 28 a day and 13 at night, but we can get up to 40 like a lot of those inland cities. Winters are quite cold, but with an average of 12 degrees a day and about 2 degrees at night. The gardens was planned back in the 20s and envisioned by the designers of Canberra, the Griffins, and then slopes to Black Mountain were chosen by Charles Weston. Construction was delayed until the Second World War, and the first plantings started in 1949 and in the 50s, and development really ramped up in the 60s. We specialise in Australian native plants, and that's all we grow. We hold the largest collection of native plants in the world, Australian native plants in the world, sorry. Our living collection has about 83,000 individual plants, which represents over 4,300 species and about a fifth of Australia's native plants. Now we have a big problem. It's called Armillaria luteobubalina. Armillaria species is found worldwide and comprises approximately about 40 species. Armillaria luteobubalina is endemic to Australia and is a major pathogen at the ANBG, and it is endemic to Black Mountain. It's also known as root rot or Australian honey fungus. The Amilaria is the Northern Hemisphere Armillaria, and that is also known as honey fungus. But that's not our problem. We've got a specific one here. It is a basidiomycete, so it develops mushrooms as the fruit and body. It has a mycelium that grows through the roots and then up the trunk in right conditions. It is a root disease fungus, a pathogen and a decayer of wood, both living and dead. The mycelium infects the roots of the plants, and it inhabits the cortex of the roots, and then it'll spread up along the roots and then up the stem if conditions are right. The two photos there are of the mycelium that I've just on the stem. I took those yesterday. I just peeled back the bark just to expose the white mycelium. It needs moisture to spread up the trunks of trees. This is a photo of a stringy bark, and that provides a perfect microclimate for it to spread up onto the bark when the surrounding garden beds are irrigated heavily and regularly, as what always happens in a amenity garden. Excuse me. It's had a very devastating impact at the gardens. It's worse probably in the rainforest, in the Tasmanian rainforest gully. So we've got two pictures there on the left. They were taken in the 1970s, and the area was quite heavily forested, and then started dying off in probably about the 2000s quite severely. So the pictures on the right really show how badly it has got. It even gets in the tree ferns, and it's growing right up and has killed the tree ferns. That's a Dixonia antarctica there. And I couldn't find a photo, but we did have a photo of mushrooms coming out of the top of the Dixonia. It would look quite spectacular, but it just shows how devastating the disease is. It's a real shame, that one. Now, I've got a picture here of what used to be called the Diures Lawn. So it was an area that was known to be an area that trees wouldn't grow in. It was always a grassy habitat with orchids in it, hence the name, and that was chosen as a site for the rock garden. It's now surrounded by a severe Armillaria infection, which is probably there all along. So what we're thinking is the Armillaria was present before the gardens was built, and in fact built around it, and it started showing up once we started changing the habitat and the horticulture there by a lot of watering and moving things around and changing things. And I think it's been quite devastating all around that area. So, but what is interesting about it is that not all plants are knocked off. This is a photo of a Eucalyptus scoparia. And it's very healthy. Now the photo on the left is of the Armillaria Scarring. So the scarring, the Armillaria has come up through the roots and up the trunk, but it's died off. I don't know why. It might have dried out. Conditions might not be all that favourable for the Armillaria, but the tree itself is very healthy and actively growing, and it is probably one of our largest trees at the gardens. It very much depends on the health of the tree, and I think genetics probably play a big part too in how the trees will cope with it. Healthy, actively growing plants will outgrow the rate of death of the roots. Therefore, environmental conditions play a big part, and in stressed plants, as with most pathogen plant relationships, are more likely to succumb to a disease. And with Armillaria, that is absolutely no difference in our experience. Now, strategies used to control Armillaria at the gardens, we've every year we'll remove the fruiting bodies, and that'll prevent the spores from spreading around. Mushrooms are collected and then disposed of in the annual bonfire of diseased material. So we collect all the material from any diseased plants, and we pile them up and set fire them in the winter. Other ways removing of infected material. The top photo shows we're trying to just dry it out in that case there. So we had a go at exposing all the roots as much as we could in that area, and eventually we ended up taking all the stumps and roots out. But obviously have missed some because it's come back with a vengeance in that particular area, unfortunately. So the heat, exposure to sunlight will dry it out and actually desiccate the mycelium and basically kills it off that way. We've also tried underground exclusion barriers with a root barrier. So the rainforest gully would be the classic case there. We dug a 100-metre long trench that was 2 metres deep, and we lined it with a 2 millimetre thick root barrier, and then built it back up and compacted it as we went. So far, that's been really good. I suspect we've had no instances of Armillaria in the rainforest gully at all. So we're pretty hopeful that strategy has worked. So exclusion is certainly a way to go. Biological controls. That's another strategy that we can probably try and have been trying. Use of the Trichoderma species. There's commercial preps available of that, and we apply it onto known affected roots in affected areas. And anecdotally, we think it's working, but we haven't got any good science behind it to back that up, unfortunately. But there's some very good papers that have been written on Trichoderma and how it is antagonistic towards pathogenic fungi, which also includes the Pythium, Rhizoctonium, the Verticillium, and Phytophthora. Another way is probably mycorrhizal fungi applied at the plant out stage. We dust the root balls as we plant them, and we're hoping that the mycorrhiza will actually keep Armillaria out. There is suggestion about that in some other papers. I think that'll probably work with, but the problem there also is the Trichoderma could have an adverse effect on your mycorrhizal fungi. So you have to be careful which way you go on that one. Basically, we have to learn to live with it. Got to recognise that it's virtually impossible to get rid of. It's costly to remove and dispose of because you've got infected roots in the soil. So where you put it, that's going to cost money, and then you've got to dig it out. That's also costly. So one of the best strategies is to learn to live with it. That can be done by changing the use of the areas that are badly infected. You can grow short-lived herbaceous material. We're trying that below the rock garden with our Asteraceae garden. So hopefully we'll just outgrow it that way. So far, so good. None of the Asteraceae plants have shown any sign of the Armillaria, the short-lived ones. Some of the longer-lived ones could possibly have it at the moment, but yeah, we're trying to improve the horticulture in those areas to get that on going. It's important to secure your valuable collections elsewhere. That's probably really, for Botanic Gardens, that's what you've got to watch out for, because that's what we're doing, is looking after quite a valuable scientific collection. So you have to keep moving around and make sure you're not planting in really affected areas. Alternate use of space, you can grow lawns or develop buildings there instead. But that's just changing the use of the areas. Important not to grow large trees in areas that have the potential to get reinfected. That'll give you a lot of grief down the track. We do have Eucalyptus regnans in the gully that might have it, so we have to be really, really careful on that and watch that one. That's where we're trying mycorrhizal fungi on that, pulling back the roots and busting the existing roots. A bit early to tell yet. I think it's had a good response on the foliage on that. So yeah, I think that will probably work. Basically, alter the horticultural practice of infected areas to benefit plants and not aggravate the Armillaria. Be careful not to overwater, and encourage good healthy plants that outgrow root there. And use containers to grow plants in. This is something that we've been trialling a lot at the gardens. That's that photo there is 2 concrete pipes stacked up on top of each other, and we've painted them with a rust colour to disguise the concrete. And there's some more we haven't painted. We've planted some, I think there's Eucalyptus caesar in there, and that garden bed is affected. So we put yeah, we've lined the base of those to prevent the roots coming down into the area, so that the infection won't rise up to the roots. So we've chosen small trees there that they will live in those containers quite happily. And now I've been asked to also talk about Myrtle Rust at the ANBG. That's sort of lead into, I think, the next session. We've actually had Myrtle Rust at the gardens. Our first outbreak was in 2016. It persisted over a few years and only on a few plants, but has since disappeared completely by the last season. I suspect that the last summer may have been too hot for it to sustain over the time. Plants affected are mainly Homoranthus species. That's Homoranthus flavescens at the top, and a Calothanus species. That's Calothanus aspera at the bottom, and Rhodania. I think the Homoranthus and the Calothanus were sort of a new recording too. Thank you. That's about it. If you have any questions, please send them through.
Great, thanks for that, Phil. We have quite a bit of time for questions. We've had a few come through. So what we might do now is we'll get straight into some questions, but remember, keep popping through those questions into the chat box and we can ask them. So we'll jump straight into the first question. So Phil, do you pre-treat plants in the nursery to give them a better chance at resisting Amilaria attacks?
We try and grow good healthy plants in the nursery, but we're not inoculating them in the nursery at this stage. We will be discussing that down the track, and hopefully we'll probably do that. But there's a bit of evidence that will work.
What are the root barriers that you've put in place? What are they made of?
Oh, they're plastic root barriers, so they're 2 millimetre thick. They're commercially available. This was a green one that we use, a very, very stiff one. I can't remember where you got it. We got a big roll of it, but it's quite thick and very rigid and quite effective.
Yeah. Okay. Okay, so it seems that plant health and optimum conditions, including addition of biota, builds resilience. The question is just wondering if there's any non-antidotal evidence or measures that we can draw upon.
Not from our area. We're quite a small unit. We've only got 12 staff, so we do everything there. So collecting that sort of evidence is, we just can't do it at this stage. It'd be really nice if somebody out there could sort of pick up on that and help us out even. It would be really good.
Yeah. Okay, so there's a question about Myrtle Rust and where it's spread. So I guess how widespread was it within the gardens? And is there reports of it being in Canberra in the wider community past the gardens as well?
No, I don't think it is in the wider community in Canberra. With our sort of sphere of influence, we've talked about it a lot with the friends of the gardens, etc. And no one has ever reported it to us. So I suspect it probably isn't. It wasn't widespread in the gardens at all. In our Myrtaceae section along the main path is where we found it. There are a couple of plants in the rainforest gully, but it didn't spread within the rainforest gully onto other myrtaceous plants that were right next to the affected plants. So yeah, there was only a couple of areas that were bad. One really bad area was where the proposed conservatory site is. And we cleared that quite recently, or 2 years ago, we cleared that in preparation for the conservatory. So all those plants got removed and then they were burnt at the time. So no, not widespread.
Great. We have, just pulling up some questions. Have you tried phosphonic acid application, and if so, what were the results?
No, we haven't. I have looked into it, but I think you've got to have a very committed program to do it, and we just haven't ventured down that path. We'd rather look at other methods. So no, I've got no idea of the results on how that would be.
Okay, another question. So does Armillaria have beneficial symbiotic relationships with any other species planted at the gardens?
Nothing planted at the gardens, but it has been noted that the orchid Gastrodia does grow with Armillaria, but I have heard somewhere, I can't remember where, that there might be a relationship with the potato orchid, but no harbored evidence on that. But it's I don't know of any other symbiotic relationship. Yeah, it's a nice little food source too for other critters, I reckon. So something munches away at it, because you can be looking at it in the roots and it's suddenly gone, disappeared, and lots of little bugs and stuff fall out. So yeah, could be something eating it.
Great. Okay. What practices have you put in place to prevent the movement of the pathogen on tools and footwear, etc.?
Okay. It's not soil-borne, so it won't be in the soil. It'll only be on root material. So, you know, we do try and be hygienic with our tools and clean off the soil from the tools, but that's more for Phytophthora and other soil-borne fungi. But Armillaria will only be on the roots, so it's the movement of infected plants and infected root material, so that if you move those around, you will transport it around. So we avoid digging up plants. We will not transplant plants from an infected area and put them elsewhere. That's the main golden rule there on that one.
So do you have a map of the spread of Armillaria in the Botanic Gardens, and how quickly does it seem to be spreading throughout the gardens?
Yes, we do have a map, but I think the spread is quite slow. Yeah, it's a hard one to quantify. There's only one new location that I know of that's popped up. So normally it'll just, the edges will just expand. But some of those edges haven't expanded ever. They've always been in one spot, and they've never gone on further than that. And other areas they have moved. So the Tasmanian gully is probably the #1 example of a severe spread, where it spread right through. So we knew early on that it was on one side, and I think probably take about 15 years and it got to the other side of the gully. But it may have already been there, but we just didn't see any symptoms. So I think it does sort of creep along quite slowly. And the new location of the outbreak, I have no idea how that occurred, because we're pretty good at collecting the mushrooms and disposing of them, and the spores don't go very far anyway. So I don't think it's a spore. I suspect it may have been an infected root material that was probably used as fill for a building when the building was built. You know, that would have been 35 years ago. And because it can lay dormant in the soil for up to 80 years. I think Glenn Kyle, when he did some research, was doing research in Western Australia on that, I think he demonstrated that. So that's potentially what happened. Was a piece of infected root that was in the soil, and other plant roots grew into it and then reactivated.
Great. A little bit more of another question on the positioning of root barriers. So with your experience, where's the best place to put those root barriers in?
It's really hard with root barriers, because in one area we actually bisected the infection zone. We thought we were right outside, we were on the right edge and outside it, but we must have bisected it because it popped up on, well away from, well, it affected a tree that was about a couple of metres away from the root barrier a number of years later, and there was no root movement across that barrier. So we strongly suspect, or when we were putting it in, we thought we might have cut the infected site in half. So you really got to know the extent of your infection and put your root barrier well and truly outside that, because it's a waste of time and resources if you just put the root barrier in the middle of it and you've got infection on both sides of it then.
Yeah. So we have a question about susceptible plants. Do you know if Agnes is susceptible to Amilaria? So Agonis.
Oh, Agonis.
Oh, yeah,
susceptible to Myrtle Rust, I know that. Yes, it will be susceptible to Armillaria. I think everything is pretty well. I've seen it on bracken fern. I've seen it on Dixonias. I've seen it on conifers, on just about everything, really. Yeah.
So I guess the follow-up question is, should sprinklers be kept off the trunk of these plants?
That would help, yes. Reducing the water and just drying out around the base of affected plants would be a really good way of controlling it. Yes, yeah, drying it out would definitely help. So with that picture of the Macrohinca, the stringy bark, that was I think that was recorded up to about 2 metres at high, but that was surrounded by a lot of shrubs. It was on the edge of the rainforest gully, but across the road. So there was no root-to-root contact into the rainforest gully, but the conditions were similar. So it was a very wet, moist area, and the stringy bark, it just held the moisture, and then the fungus went straight up the side of it, up to about a 2 metre mark, I believe. Yeah.
Okay, great. We might leave it there for today. I'd like to thank Phil again for his time today and giving a great presentation and also answering so many great questions that came through. Just before everyone signs off today, it would be fantastic if you could just quickly fill in a quick poll for us. And I would also like to give you a bit more of a rundown on some of the activities that are happening within the Botanic Gardens Biosecurity Network over the next few months. So over the next few months, from September on to November, the network will be conducting a series of surveillance blitzes. So what these will be, there'll be 3 surveillance blitzes. One will be looking for Myrtle Rust, another will be looking for brown marmorated stink bug, and the other will be looking for the presence of tree of heaven. Now, what we're going to encourage all of our network to do is to go out within a week period, within that month, and to go out and look for those pests or plants, and then to report anything that you see through My Pest Guide Reporter. So these blitzes will be promoting through our Facebook page and our website. So keep following our Facebook page and our website over the next few weeks to start to get some information about these blitzes. And we really hope that you can all join in within these blitzes as well. Now, finally, we have our very final Botanic Gardens Biosecurity webinar coming up on the 26th of August. And this webinar will be on Myrtle Rust, and it will feed right into the Myrtle Rust Blitz. So this presentation will be done by Angus Carnegie from New South Wales DPI. And he'll be just talking a little bit about when it was first detected, and talking a little bit about how to identify it and where to look. So join us in 2 weeks' time to learn more about myrtle rust and how to keep an eye out for it. Okay, so we've had most of the polls come in now. Once again, I'd really like to thank Phil for his time today. It's great hearing experiences from each of the Botanic Gardens. And I think it's been a very, very useful webinar today, and I've definitely learnt a little bit about the Botanic Gardens experience here in Canberra. So with that, I think we've almost had all attendees answer the questions. I think we'll call the webinar closed. Thanks again to all of our online attendees, and thanks for joining, and we hope to see you in 2 weeks' time.